In this video, we're going to talk about the second line of the innate defense. So even though we're going to talk about cells, this is going to be the second line of the defense. First line is, again, the barriers or the walls and the chemicals that they produce or the enzymes that they produce to kind of prevent microbes and pathogens from getting over the wall. The second line of defense is still cellular, so it's going to be, again, don't get it confused with the third line. So it's going to be cells, but again, they're non-specific, meaning it doesn't matter the pathogen that's coming over, it's going to fight against it. So they get over the wall, and you have cells that are ready to fend against any of those microbes that get over the wall. It doesn't matter, again, whether it's the flu virus or a staph bacteria, they're going to fight against it the same way. So they get into kind of the deeper tissues. Still innate, still non-specific, but more cellular in defense. So this is going to be our phagocytes. Again, phagocytes are going to be eating cells. So what they do, specific white blood cells that ingest or eat and then digest any of those foreign invaders. So again, it's non-specific, but they're going to take these pathogens into their cells if they're not already intracellular pathogens. They'll take them in the cell, they'll use their digestive enzymes to digest it, and they'll spit out kind of the waste products. So first off, we're going to talk about kind of the white blood cells again. So we're going to talk about neutrophils, macrophages. These are also going to include natural killer cells. Natural killer cells are going to be a type of lymphocyte. So again, don't get it confused. These are going to be non-phagocytic, so they're a little different. So we're going to talk about the phagocytes, which is those white blood cells that will fight against anything. Natural killer cells are different, and again, they're non-specific. They'll kill anything, but they're non-phagocytic, and we'll talk about how they kill their pathogens when we talk about them. So the second line of defense that's not cellular, but still just as important, is actually inflammation. So we'll talk about that as well. So often we think inflammatory response is bad. So you have swelling, you have redness, you have pain in an area. That is actually your body's way of trying to fight off the infection. So we'll talk about the inflammation as well, probably in the next video. We also have antimicrobial proteins. So these aren't cells, they're protein molecules that help, again, defend against all sorts of different microbes. And they're good with communicating between kind of different parts of your immune system, and they can even kill microbes themselves. So we'll talk about those coming up later. And we also will talk about fever. Is another thing that we hear fever and we're like, oh no, that's bad. But it is bad if it gets too high. Yes, that's very true. But a normal low-grade fever is actually your body's way of trying to stop whatever's going on. So it is actually a positive thing if it's slight, because it's part of the way your body's fighting off any type of infection. So first, talking about those phagocytic white blood cells, the first ones are your neutrophils. Remember, these are your most abundant, meaning the most numerous phagocytes. So we see the most of these. Remember, 60 to 70% of your white blood cell count in your blood smear is going to be 60 to 70% neutrophils. They circulate in the blood. They'll also go into your connective tissue. Remember, connective tissue is your widest range of tissue. And they are going to often be fighting against bacteria. Now, again, it doesn't matter the bacteria. So it can be staph, it can be strep, it can be mycoplasma, it can be any type of bacteria. But they're going to oftentimes fight against bacteria. They die fighting. So there's most numerous of them, but they're usually the first to die. They eat and consume some bacterial infection, and they only eat a couple of things and then they die. So oftentimes you'll see kind of degenerated neutrophils or dead neutrophils, just because they die fighting. So there's the most numerous, but they die quickly. So oftentimes if you see pus, it can be a lot of dead neutrophils. They do become phagocytic. Again, they're eating whatever they are exposed to. Um, they can also produce what we call a cloud of bactericidal chemicals, meaning bacteria killing chemicals. They have kind of lysosomes, and what happens is this can release, again, digestive enzymes and have kind of a killing zone around it. So even though they can only eat a couple times, they can actually help kind of produce a little zone of death around them that'll kill even more bacteria. So they can kill several bacteria at a time. They can also produce a protein that can actually poke holes in the membrane of a pathogen. Oftentimes, again, it's a bacteria. So if you have a bacteria, unicellular organism with a cell membrane, if you poke a hole in its cell membrane, it's kind of the same process that happens with our cells. All of a sudden, water's going to flow out and you're going to have this shriveled mess. So if you're poking a hole in the cell membrane and it's no longer separating the intercellular from the extracellular, then you can have a destroyed cell. So that's another way that they can kill bacteria or kill pathogens. Eosinophils, oftentimes you're going to see in your mucous membrane, so the kind of the opening, like your oral cavity. These guys like to fight against parasites, so a little bit different. They also help against allergens and other pathogens. So things like tapeworms and roundworms are large parasites in your intestines, intestinal parasites. So they can actually kill roundworms and tapeworms in the intestines by producing kind of oxidative chemicals. So superoxide and hydrogen peroxide and other toxic proteins. So, hydrogen peroxide, we pour it on something, it's very destructive, and it'll kind of destroy cell membranes and denature proteins for hydrogen peroxide. So eosinophils can slowly kind of destroy these larger parasites because they're worms, and destroy them using this kind of mechanism. They are also phagocytic in the fact that they eat antigen antibody complexes. Now, we're going to talk about these a lot more when we get to the specific immune system. But what happens is antibodies, say you have an antigen, which is anything that causes an immune response. So remember that anything that causes an immune reaction is an antigen. So it's not always just bacteria. It can be um, it can be pollen or um, any toxins, anything that's causing an immune response. And antibodies, their purpose is we usually draw them as Ys. And again, we'll talk about them a lot in later videos when we get to the specific immune system. But what antibodies do is they surround, oftentimes, they'll surround the antigen. And we saw this with our blood typing where they cause a clump. So antibodies here are going to surround the pathogen and make it so that it can't function anymore. It can't attach to anywhere. It can't produce harmful chemicals. It can't reproduce. So it kind of neutralizes the threat. So you can think of it as kind of it arrests the criminal. Then eosinophils can come in and they recognize kind of that this is abnormal. Again, they're not specific. So this could be any type of bacteria, any type of fungal infection, anything. But eosinophils will come in and they'll recognize that that's antibodies want us to eat it and they will consume it. So this entire thing with the antigen, the guy in the middle, with the surrounded antibodies is called an antigen antibody complex. So that's what it'll come in and eat. Eosinophils in their kind of help against allergens as well. It's going to limit the action of histamine, which histamine is going to be our major inflammatory chemical that we'll learn about in later videos. So that is going to be limiting the action of histamine and other inflammatory chemicals. So this is actually where it comes in that's going to decrease inflammation, which it can help against allergies, because if you're decreasing kind of inflammation against allergens, that'll help with kind of the symptoms of allergies. Basophils are going to be more not the phagocytic, they're still non-specific, but they're going to help kind of communicate with other leukocytes. So they'll help bring in and attract other leukocytes, meaning it'll attract neutrophils and eosinophils. So this is the guy that's getting on the microphone and yelling, help, help. And it's sending out a chemical. Leukotrienes are going to be one specific chemical that's going to help bring more help to the area. So it's going to say, I need more help, or this area is damaged, so neutrophils and eosinophils need to come to the area. Basophils increase inflammation because all of those little granules that we saw, those black, blue granules that we saw on basophils, a lot of them are containing histamine. So I just mentioned histamine with the eosinophils. Histamine is inflammation. It's our major inflammatory chemical. So when you take an antihistamine, when you have a runny nose, you have puffy eyes, itchiness, all of that, that's part of inflammation, and that's caused by histamine. So when you take an antihistamine, it's fighting against the effects of histamine. One effect of histamine is a vasodilator. Now, we learned in the blood vessel component that this is going to increase the diameter of the blood vessel. If you're increasing the diameter of the blood vessel, then you're decreasing resistance. So you're increasing blood flow. Why is this partly important? Again, we'll talk about more about inflammation later. Well, more blood flow means more area for leukocytes or white blood cells to come to the area. So there's more room for all those neutrophils and eosinophils to come. We also have a chemical called heparin. So heparin is going to be an anticoagulant, meaning it's going to not allow clot formation. Now, this is important because if you have, say, a wound or you have an infection in some area, you don't want blood to clump and clot. So you don't want those platelets to come in and clot up the area, because you want white blood cells to be able to move freely to get to that area. So if you're clogging the way and making clots, then leukocytes or white blood cells can't get there. And that's what we want. We want our white blood cells to get to an area that is infected. If you hear a thing called mast cells, now this is different than a basophil. These are going to be in connective tissue cells, so it's actually a connective tissue cell. So mast cells are going to be similar, but they're not going to be in the blood. They're going to always be in your connective tissue, but similar kind of effects. So lymphocytes, again, we're going to have three categories of lymphocytes. These we're going to talk about later because these are your specific immunity. We are going to talk about natural killer cells. They are a type of lymphocyte. that you can only recognize again kind of with specific tests, but natural killer cells are non-specific. So these are going to be different. They're also non-phagocytic, meaning they kill in a different way. And we'll see next. So when we're going to talk about B cells and T cells, so T lymphocytes and B lymphocytes, they have a specific match that they're looking for when they're circulating. So we have in circulating blood, 80% are actually T lymphocytes or T cells, 15% are B cells, and then 5% are natural killer cells. A lot of times B cells like to hang out in your lymph nodes. That's why there's less circulating. There's still a bunch, but they're not circulating in blood, so they're not traveling in blood all the time. Again, natural killer cells, innate or non-specific. Remember I mentioned this, monocytes. So we looked at monocytes when we talked about blood. Once they move into connective tissue, then they become macrophages. You're going to hear about macrophages a lot in this section. This is our major phagocytic cell, and again, I call it the Pac-Man of the immune system because all it wants to do is eat. That's what it does, eats or phagocytize. We have two types of macrophages just based on if they're stuck to something or not. So free macrophages are going to be oftentimes in loose connective tissue where they can travel around and look for pathogens. So they can look for food. We also have the lazy macrophages that are going to just wait for the pathogens to come to them. So we have things in our alveoli, so the air sacs of your lungs. We have them in our liver, and specific microglial cells in the central nervous system are a type of macrophage. So most of your macrophages are going to be free, but we do have ones that are the lazy ones that are in specific areas that'll just hang out until the pathogen comes to them, mainly protecting the lungs, major part where we want to protect ourselves. Um, Anything pathogens getting in the lungs can be very dangerous, and the liver, same thing. So phagocytosis, just remind you about the process of it. So what happens is the phagocyte recognizes kind of the sugar signature that's on the outside of the cell that it's abnormal. So remember, you have a glycocalyx or a sugar signal that's you. So this may tell everyone that that's your cell. Well, phagocytes can recognize, okay, this is a different signature. So this is an abnormal cell and needs to be destroyed. So it recognizes that kind of sugar or carbohydrate signature. What happens again is the cells will reach out with what we call pseudopods or false feet, and they'll give whatever they're eating a hug. So they'll enclose or kind of surround the pathogen with its little arms or false feet, pseudopods. Stick to the pathogen and then bring it inside the cell. So it'll complete the plasma membrane totally around it and bring it inside the cell. Once it's inside the cell, so it's inside a vesicle because that's what we've done. The plasma membrane is completely surrounded it. That's called a phagosome. Then remember, it'll fuse with a lysosome. Lysosomes have digestive enzymes that are naturally in vesicles inside our cells. So lysosome will come along and it'll fuse with the phagosome. So then you get a phagolysosome. So then the enzymes digest the particles. Anything that we can use and recycle, the cell is going to keep because why waste it? And everything else that's waste products is going to be exocytosed or released from the cells. So we've kind of digested all and we keep our digestive enzymes and recycle everything. So we digested all the product and we don't need it anymore. So then we refuse with a plasma membrane and release all the particles out of the cell. So just looking at it again, better picture. So the phagocyte adheres, and we make our pseudopods and eventually engulf it and completely and surround it with cell membrane. So you have phagosome, a lysosome comes and fuses with it, digests it with its enzymes, and then we exocytose or release the digested material that we can't use. So some pathogens, when we bring them inside, can't or do not kill or can't be killed with our lysosome enzymes with some weak acid added to it. So macrophages, again, are major phagocytic cells of our immune system, can produce what we call respiratory burst. It releases what we call free radicals, which are very damaging. And they can also produce oxidizing chemical. Hydrogen peroxide is another way. So that's why you put hydrogen peroxide on a cut and you just put it on once. You don't want to repeatedly put on hydrogen peroxide because it can actually be damaging to your tissues that are trying to heal. So these are going to be damaging to the proteins, and these can help kill the whatever pathogen that's added to the macrophage. So it actually increases the pH. If they are bacteria that like acid, then we make it more basic. So increase or make it more alkaline. And we can also increase the osmolarity of the phagolysosome, so we can kind of bring more solute in the area. So things like sodium can be damaging to some bacteria. So we'll use other mechanisms besides just the lysosomal enzymes to kill some pathogens if they're kind of able to withstand these enzymes. Again, I mentioned we're going to talk about natural killer cells. Again, a type of lymphocyte, but it's going to be non-specific. And they are non-phagocytic. Right there. So they look and travel around the blood and lymph. Now, again, they are different in that they're not killing against extracellular components. They are fighting against intracellular components. So we have pathogens that either like to stay in the tissues or the bloodstream, or we have pathogens that actually want to live inside the cell, and that's going to be most of your viruses. So if this natural killer cell comes upon a cell that's in either cancerous or infected with a virus, then it's going to kill it. And it can kill it before, again, your adaptive immune or your specific immune response is activated, because we'll talk about the specific and how it fights later on. So how does the natural killer cell kill the cell that's infected? What happens is it pokes holes in the cell membrane. Now, this is going to be your own cell, but it's going to recognize that this cell is going to be infected. So the cell is just going to say, kill me, because I am taken captive, I'm kidnapped, so get rid of me. So what happens is the natural killer cells put what we call perforins or holes in our target cell's membrane. So we poke holes into it. So holes in general are dangerous. Again, the plasma membrane needs to be intact. Make sure that there's kind of appropriate movement. If you have holes in the plasma membrane, mainly water is going to travel in and it's going to be able to freely move in. So you're going to have a cell that, or water is going to freely move out. So you're going to have a cell that's going to be damaged in that way. Added to it, you add enzymes called granzymes that are um, going to be injected into the cells, and they help induce cell death or apoptosis. So not only do you have the poking holes in the membrane, but just to make sure that it's really good and dead, it's going to go inside the cell and have cell death occur as well. So you see natural killer cells are circulating, but oftentimes they'll hang out in the spleen, our major lymphatic organ, larger lymphatic organ, our most numerous lymphatic organ, red bone marrow, and it also circulates through blood. So again, these are non-specific, and they're going to fight against intracellular pathogens, intracellular. So we're going to see natural killer cells are going to kill the same way as T lymphocytes. Again, I'm going to talk about it again. But T lymphocytes are specific. So natural killer cells will kill anything that it recognizes as infected. So again, it doesn't matter if it's coronavirus, flu virus, any type of virus, it's going to fight against. T lymphocytes will only be a specific strain of the flu virus or a specific strain of, good example is COVID. So T lymphocytes are specific, natural killer cells, non-specific.